Hello and good afternoon. It's 2 p.m. UTC-4. I am pleased to welcome you to another symposium of the second virtual meeting of Systematic Paleogeography and Evolution that we kindly nicknamed SBE Meeting 2021. And this is the first talk, the keynote talk for the symposium entitled Current Methods and Applications of Big Data Phylogenetics. Is with an immense honor that I introduce to you Dr. Rebecca Dikow. She is a research data scientist and leads the Smithsonian Institution Data Science Lab, which is part of the Office of the Chief Information Officer. Since starting the Data Science Lab in 2016, she has been conducting biodiversity research using genomics, informatics, and machine learning tools. More recently, the Data Science Lab has begun working with researchers studying digitized collections and archive data outside the biodiversity sphere and strives to collaborate with scholars all across the Smithsonian. The Data Science Lab also provides support for researchers using high-performance computing clusters and training in data science and bioinformatic tools. She is also an affiliated faculty member in the George Mason University School of Systems Biology and the Smithsonian Mason School of Conservation. Thank you very much for joining us. Dr. Dikow, the ball is in your court.
Okay, hopefully you can see my slides. Please let me know if not.
Works great.
Great. So I'm really happy to be here today. I thought I would talk today about some of the breakthroughs that have happened with genome-scale phylogenetics over the past 10 to 15 years, and also the continued challenges. So not everything is solved, and big data definitely don't solve all our problems just yet. So just a little bit about the Smithsonian for anyone who hasn't been there or isn't familiar. The Smithsonian is a museum and research complex in Washington, D.C. We have 19 museums, 9 research centers, and a zoo that are all part of the institution. And I just have some pictures here. In the middle is the Smithsonian Castle. We call it the castle. It's kind of the headquarters. And then on the top left is the National Museum of Natural History, where I do a lot of my work. On the top right is just a photo from the insect collections there. And then on the bottom left is a photo of the Smithsonian Tropical Research Institute in Panama, which is another outpost of the Smithsonian. And I collaborate a lot with researchers there as well in genomics and some systematics. And in the bottom right is a picture of our National Zoo. They also do a fair amount of genomics at the zoo and are a great collaborator. Within the Smithsonian, our group, the Data Science Lab, is part of the Office of the Chief Information Officer. So that's kind of the IT organization of the Smithsonian. And we're a small research group where we do research at that intersection of computers and collections data. So we do a lot of genomics research, as well as some machine learning work using collections data. And as Dennis mentioned, we help support the high-performance computing cluster and do training, mostly using carpentries style training. So part of that work, to me, is really interesting and exciting because we get to help a lot of researchers with some of the problems they're having running bioinformatics software tools on the cluster. So beyond my own interest in research, I get to kind of see through other people's work how, lots of different software programs and how they're using them. And so I thought that I would kind of use that today as a way of talking about how genomics has become a big part of phylogenetics. So as I'm sure you're all aware, improvements in DNA sequencing technology and analytical techniques over the past 15 or so years have made it possible to generate huge genome-scale data sets for really diverse taxa. Sorry. Oops. But there are a lot of challenges. And so I'll talk about 3 kind of major sets of challenges. First, in study design, whether you have a reference genome available, what kind of quality your DNA or RNA you need for your particular application, how suitable is your data for your question of interest, and cost. And then in data processing, there's all sorts of new challenges. There's the potential for contamination within your data. There's the question, do you have access to the sufficient high performance computing resources that you might need to analyze your data? And also there's this issue of complicated and constantly updating software workflows. And then for phylogenetic analysis itself, once you have generated these huge data sets that take a ton of work to generate, how do you subset and analyze the data in a way that you feel like you're going to be able to answer your phylogenetic question? And this also may need significant high performance computing resources. And then how to assess confidence in your hypothesis. So I'll talk about all of these things. Unfortunately, there's no maybe good answer to any one question. So it really depends. The answer is it depends. So, right. So the approach you want to use really depends on what taxa you're interested in. What is the divergence time of those taxa? How many samples do you think you want to be able to put in an analysis? What's your budget? What kind of sample quality do you have? Are these museum collections? Are they freshly collected tissues? Are they really, really tiny insects where you won't be able to get a huge amount of DNA? And what are the computational resources? So I don't have a lot of pictures of cool organisms in my talk because it's mostly about methods in bioinformatics, but I chose some images of collections objects from the Smithsonian to kind of illustrate a few points. And this, to me, just, it's kind of a juggling act, a balancing act. I don't know how you want a cost-benefit analysis, however you want to think about it. There's so many things to consider when starting out on a genome scale project. And I just thought I would work through an example from birds. So in the past 12 years, so 2008 to 2020, here are some of the kind of big papers that have come out looking at phylogenies of birds and their increase in data over time. So this paper came out when I just started grad school, and it was considered a really big paper. This is a really important paper. And you can see they included 169 species and 19 loci with 32 kilobases of aligned DNA. And we can look back at this 12 years later or 13 years later now and say, wow, that's not a very large phylogenetic matrix. But the only reason we can say that is there's just been so many advances in terms of analysis techniques and sequencing throughput that have made that possible. In 2013, the first UCE paper came out, so ultra-conserved elements, target enrichment data, and we'll talk a little bit more about that later. And in this case, look at the number of loci. It's gone up by 10 times, and fewer taxa in this case, but the number of loci is just huge, and this really represented a big change in the way people construct phylogenies. In 2016, this kind of bird genome effort to sequence lots of bird genomes really started taking off. And this paper was published with 48 whole genome phylogeny with 48 species of birds, which is just a tremendous amount of computation and also data wrangling and decisions about which parts of the genomes to include. This was a really big paper. And then this just last year, a paper with 363 species with whole genomes was published using whole genome alignment to come up with the data sets. Again, this is, many taxa don't have enough genomes to do this kind of level of analysis, but this is definitely the way things are heading. You'll also notice that the earlier papers I mentioned still had a lot of authors, but as we go on, these papers have hundreds of authors because this is not the kind of work that small teams can do. So what are these big breakthroughs? The breakthroughs are in both the long read sequencing becoming both more available to people, and also the chemistry behind these PacBio and nanopore techniques has improved so much in the past couple of years. The first time I personally worked with PacBio data, a single cell on a PacBio machine would give you about a gigabase of data, and now you get like a terabyte of data from one cell using the PacBio Hi5 chemistry. So there's just a huge amount of efficiency change, and also the cost. It's still somewhat expensive, but it has gone down so much per base pair. And this allows us to sequence, repetitive genomes, plant genomes, for example, in particular, really long reads have really made it possible to sequence these genomes and to generate contiguous genomes. And then Hi-C and other proximity ligation methods have made chromosomal level assemblies possible and within the realm of within reach for many taxa, whereas before that was really only possible for projects that were very, very well funded and often had, were related to, mammal genomics and human health or have some other application, whereas now it's kind of much more in reach for biodiversity researchers. And also for not kind of full genome stuff, but for reduced representation genomes, the ability to multiplex hundreds of samples on one Illumina lane again, the improved throughput of different Illumina machines has really made it possible to generate very large data sets fairly cost effectively. In a recent review that came out this year, looking at how long reads have really changed in insect genomes and how long read technology has really been taken up by that field, you can see this plot on the left showing the number of genomes, and in light blue is kind of the expected based on the number of species in an order. You can see that Diptera are overrepresented, probably because of Drosophila being a model system. Coleoptera are underrepresented. But what I wanted to point out is in panel C, you can look at the yellow being the long read genomes, and just over the past 2 years, those have increased just so much, and the contiguity of the genomes, the contig N50 on the left axis, has just been increasing so rapidly. So huge strides are happening, but you can also see that there are many orders that there are very, very few genomes still. So for a lot of biodiversity researchers, this problem of not having a reference genome is still not solved. So I thought I would talk today about the kind of five of the major methods for generating phylogenomic data sets, and then talk about each of the challenges in reference to those methods. So just to introduce them in case you're not familiar, RADseq is a restriction site associated DNA markers. So you use restriction enzymes to attach to your DNA and sequence loci based around those restriction sites. There's genome skimming, where you would do low coverage shotgun sequencing, not to assemble a whole genome, but to potentially assemble loci that you could use as phylogenetic markers. Then there's target enrichment, which UCEs or ultra conserved elements are part of this larger target enrichment bin, which also you could do exon sequencing. And what this means is using probes to capture loci of interest. So you design your probes based on some reference genomes or transcriptomes and capture loci of interest and use those as phylogenetic data sets. I'm just moving around the picture so I can read my slide. So for transcriptome data sets, you use RNA-seq to assemble sets of transcripts that you could then use as phylogenetic loci. And for whole genomes, it's also shotgun sequencing, like in genome skimming, but deep sequencing, so that you can assemble the genome de novo and compare them to one another, most often using whole genome alignment software such as Cactus. So let's talk more about the challenges. First, in study design. So what factors do we need to consider when designing a phylogenomic project? So the first one is reference genome availability. How closely related is the closest reference to your taxon of interest? For some groups, there are many references. For example, birds is now really just, there's genomes all the time in mammals. I mean, I would say many vertebrates, not all vertebrates, because some vertebrates have very large genome sizes. So there's been much less sequencing in those areas. For your technique that you're interested in, do you have to sequence your own reference genome? That's something that you kind of want to know at the outset to know how to resource your project. And then are there available probe sets? So there, for UCEs, there are available probe sets for a lot of different taxa that if you can take advantage of those, you won't need to worry about designing your own probe set or sequencing your own reference genome. So for RADseq, a reference genome can be really helpful for mapping loci and determining homology, but it's not required. There are methods that don't require reference genomes. For genome skimming, again, it can be really helpful for understanding, for mapping reads or mapping your loci to a reference, but it's not necessarily required. For target enrichment, if you're developing your own probe set, it's very useful to have a reference genome or at least a reference transcriptome. And then for a transcriptome kind of phylogeny, it's also helpful to have a reference genome for homology detection, but not necessarily required. And whole genome, yes, you need a reference genome. So the next challenge in terms of study design is understanding the kind of sample you need. So do you need really fresh material? How should it be preserved? And what about museum collections? Can you use museum collections for this particular technique that you're interested in? For RADseq, you can be successful with a low amount of input DNA. The same with genome skimming and target enrichment. I am involved in a target enrichment project in composite plants, and we have used some museum specimens and we've been successful. The capture efficiency isn't quite as high as it is with freshly collected samples, but they're definitely usable for the phylogenetic analysis. For transcriptomes, you need very freshly collected material, flash frozen or RNA later preserved. This is kind of a hard one. You really can't get away with older material to generate transcriptomes. And for whole genomes, depending on the sequencing technology you're using, you often need high quality DNA and a lot of it. This is changing a bit. So there's kind of low input DNA chemistry for PacBio that is making it more possible to work with organisms with a smaller amount of DNA. If you're doing just Illumina sequencing, you don't need a ton of DNA. The problem is that in a lot of cases, you're looking for a more contiguous genome, and so you do need some sort of long reads. And in that case, you do need very high quality DNA. So the next challenge is kind of what is the phylogenetic scale? How do you know if your data are, the data you want to gather will be suitable for the scale of your question? So what is the phylogenetic scale? How many taxa do you plan to sample to see if that's kind of in line with your budget? And then what is the divergence time? So for RADseq, techniques are best for within species, and you can go a bit above that, beyond that, but there is a limit. So you are definitely not able to look at deep divergences with RADseq. Genome skimming, again, can be successful at both shallow and deep divergences, but there is kind of a homology detection step that is really important in order to make sure that you're looking at comparable loci across your taxon sampling. So that that step is essential in order to make sure that you are able to compare things across a deep divergence. And target enrichment can also be really successful at both deep and shallow divergences, in particular UCE loci, because they kind of, as you go further out, they have these cores that are very well conserved. And as you extend out from those, you can still capture a lot of data that are more variable. So there's kind of a less variable region and a more variable region. And this can be really good at both deep and shallow divergences. Transcriptomes as well can work well at deep and shallow divergences. Again, this homology detection step is really important. In target enrichment, you have your probe set to kind of help with that homology step. So that's kind of it makes that technique really accessible. Whole genomes, obviously you have the whole genome, so it can be successful at all levels of divergence. But the genome alignment step, which is kind of your homology detection step, is very, very computationally intensive and yes, out of reach for many taxon samplings at this stage. we've already mentioned a little bit about cost. Cost has gone down a lot, but it can still be really expensive to generate these data sets. And I've kind of like, for restaurants, you can see like how expensive is this restaurant when you're looking online, kind of the $1 sign up to $3+ signs. Again, these are kind of guesses. It's a bit of an estimate. It really depends on, the answer is always going to be, it depends on a lot of factors, but RADseq is definitely very cost effective. You can multiplex tons of samples. It's not particularly complicated library preparation. Genome skimming can also be an inexpensive option, but depending on your genome size, you will need more coverage in order to capture those loci that you're interested in. And target enrichment as well. The probes themselves can be expensive to purchase, but you can multiplex a lot of samples, and a probe set investment can go quite far. Transcriptomes, this also depends on whether you're also interested in looking at expression, or if you have multiple tissue types for your samples, or if you're just kind of looking at a single sample from each taxon. And whole genomes, again, this can get very expensive depending on the sequencing technology and the genome size. If you're doing multiple sequencing technologies per sample, sky's the limit on how much genome sequencing can cost. Okay, so the next section of challenges I wanted to talk about is data processing. And the question is kind of what potential pitfalls exist when processing high throughput sequencing data. Contamination. This can be a problem for a lot of high throughput sequencing data sets. We are sequencing everything in your extraction. So there's just always the chance that there's bacteria, viruses, or other contaminants in your data, and cleaning those out can be a really important step. For RADseq, through the dataset assembly process, data filtering steps can really help prevent contaminants from getting into your final data set. For genome skimming, depending on which analysis workflow you're using, there may be some filtering steps. You can also use a program like Kraken, which will classify your reads as potential contaminants on your raw data to get a sense of if you have contamination before you do any assembly. In target enrichment, again, because you're matching to probes, you are doing a lot of data filtering throughout the informatics workflow, so that helps preventing contaminants from getting into your data set. Transcriptome, the same as genome skimming, you can filter your raw reads using Kraken if you, it's always a good idea to try doing that. For whole genomes, the potential for contaminants in a de novo genome assembly is pretty high. I would say if you look at assemblies on GenBank, many of them have some bacterial contamination. Raw reads can be filtered, again, using Kraken. It's a process you can also run after you run your assembly. You can filter first, you can assemble, and then try to filter again, because it can be tricky. And as I mentioned early on, that access to computational resources can be a big challenge. So genome-scale data can take up a tremendous amount of disk space, and some software needs a lot of CPU time and/or memory, these are often very scarce resources. And I've put these little disk emojis, even though we don't use those kinds of disks anymore, I don't think, but just to kind of show the kind of similar to the cost, the progression as we go down this list, you need more and more computational resources. Whole genomes in particular really can take up a tremendous amount of disk space, a lot of it temporarily. So doing a whole genome assembly, you can kind of often balloon the amount of data and intermediate files. A genome assembly could take hundreds of gigabytes or even a terabyte of RAM, depending on the genome size and the kind of data you have. So this can be really, it can be a showstopper for a student or someone at an institution without these resources. There are a lot of resources available in using National Science Foundation funded computational resources like XSEED and Cyverse that do have resources available, but it's a problem at the Smithsonian that we're worried about, is that we have lots of genomics researchers just generating tons of data. The raw data and the processed final products get put into GenBank over time, so that's at least an archival space for them, but there's just so many extra files that people are working on for a number of years. It often takes a long time to analyze these data, and we are constantly running up against disk space problems. And also, as I'm sure many of you are aware, bioinformatics software is not always easy to run, and it's not easy to install, and there's just constantly new software, which is exciting. It's a really exciting time to be in biodiversity science, but it's also, systematists are kind of increasingly expected to be really bioinformatically savvy and to understand the whole genome workflow in order to produce a phylogeny, and that's a huge challenge. The good news is that there are a lot of really good online tutorials and training opportunities. I would particularly recommend Carpentries for foundational data science skill training. And there's a genomics lesson within the Carpentries curriculum. So finally, I just wanted to talk a little bit about the kind of actual phylogenetic analysis. And again, I don't go into a lot of detail here, but there's a lot to think about. So we did all this work, first designing our study, processing the data to have a phylogenetic data set, but then there's a lot that goes into how to analyze those data as well that can be very challenging with these larger scale data sets. And these are just kind of a few of the choices that one has to make in the process of doing this work. How much missing data to permit in an analysis? Often throughout the kind of workflow that you're using to generate your data set, you'll have to make decisions about, do you want to include all the taxa for a particular locus, or do you only want to include those loci with a certain number of percentage of taxa? How do you deal with potential paralogs? Again, if you're using target enrichment with your probe design, you can attempt to only include orthologous loci. In certain groups, that's not as possible. For example, in plants where there's been lots of whole genome duplications. Should you concatenate loci or not? Should you do more of a gene tree, species tree approach? Should you analyze different kinds of loci separately? For example, if you have a whole genome data set, or should you analyze exons separately and introns separately? Or what about sliding windows of data sets across the genome? These are all different options. You can try them all, but it can be a bit overwhelming. If you have whole genomes, what about genome synteny and architecture? Can those be informative phylogenetically? And I just wanted to point out a couple of studies that I was a small part of. This one, Edelman et al. Looked at 20 heliconius genomes. So heliconius butterflies, there's a very nice heliconius reference genome. And so these are 20 genomes that were sequenced, and whole genome alignment was done in cactus. And then, just as I mentioned on the previous slide, those kind of all iterations of how that data set could be parsed were done. So let's look at all the exons, let's look at all the introns, let's use sliding windows across the genome, really just spending a lot of time with the data to understand, how it was affecting the phylogeny. And this is another paper I was recently involved in. Verone et al. Was looking at gadiforms, which is an order of fish. And this was an exon target capture data set. So I think there are about 14,000 exon data set. And this is just her analysis workflow. Like after the whole data processing, so going from raw data to alignments, this was the analysis, phylogenetic analysis workflow. So you can see there's just a ton of different analyses that we did to really kind of as a sensitivity analysis to better understand how the data were behaving, different subsets of the data were behaving. As we mentioned before, for larger data sets, phylogenetic software can require a lot of CPU time or memory. And as we add more taxa, that usually really increases the amount of resources that are needed, more so than the amount of loci. And finally, just a little bit about assessing confidence in our hypotheses. The traditional phylogenetic support measures, like the bootstrap, for example, they're often really high with large data sets for every node. And we know that that's not really what we want, right? We want to kind of understand what parts of the tree are uncertain, or where there may be introgression, or whether there, we want to actually see the lower support values to understand our tree better. So there are some newer methods for looking at concordance factors, for example, and I put the reference here. It can be really useful in capturing this kind of topological variation in large data sets, because that's something that can be really where times have changed a lot in the old days of really scrutinizing our alignments in detail to understand, to really get into the data. Now we're just looking at often these huge data sets, and we really kind of want that information to point out areas where we might really have to look deeper. So in conclusion, genome-scale data sets can be really powerful, but they're really subject to a lot of the same phylogenetic challenges that we've been dealing with for decades. They don't solve every problem. They can be kind of the next step and really exciting, but we still need to really think hard about all the decisions we're making with our analysis. But I don't want anyone to be deterred, especially students just learning these methods by the challenges of these data sets. But just kind of be aware of these factors as you're designing your study and thinking out your plan for analysis. And also finally to seek out collaborators, because as we're kind of increasing this toolkit around genomics, it's impossible to be an expert in every aspect of this work. And so, I am involved in a lot of collaborations with kind of experts on lots of different taxa. And, it's a really great experience to kind of bring all of our different expertises to the same problem. And so I encourage you all to do that as well. And I would just like to thank my many collaborators at the Smithsonian and everywhere, and a final little collections object from the American History Museum at the Human Genome Map in 1990, which is now finally, we have a complete human genome, which is really exciting. But now I'd be happy to answer any questions.
Thank you. You may still keep sharing if you want to leverage on your slides for the questions and answers. And thank you very much for this very insightful talk. That was so easy to understand, although you wrapped up a lot of very important information in there. Before we go to question and answers, I would like to tell all the participants that we have here that you may type your questions in the chat, and that we are going to keep going with questions and answers here for a while. However, please don't forget that we will have our next speaker, I might be butchering his name, Frederick Del Suc, at 2:50 p.m. sharp. He will be starting his talk no matter what we are doing here. And I will put the link for his talk here in the chat, so that if you have it ready. Okay. Without further ado, I have a comment question by Barbara Leal. She says, what would be the best approach to deal with allopolyploids, reticulate evolution, in parentheses, within a clade?
Yeah, I mean, that's definitely a big challenge. There are some software packages, which I'm kind of blanking on the names right now, but I can try to look them up, that are specifically built around plants to start, trying to clear up issues of allopolyploids within phylogenetic data sets. I know there's a lot of different probe sets, target enrichment probe sets designed around plant genomes. And so bioinformatically, you can start to sort out those loci that are paralogs after you generate the data themselves. But it's definitely a big challenge, and it's one that's definitely evolving how to deal with that.
Thank you. While we wait to see if more people type their questions, I have a few. First, you just mentioned concordance factors and the importance of maybe looking into those, and maybe at the same time looking into support values. And I noticed that perhaps this could be new to some of our audience. Could you please go into a little bit more detail about what those concordance factors are and how can they inform us better about the topology in comparison to traditional support values?
Yeah, so let me actually put the link in the chat because there's a really good blog post about how to use concordance factors. Related to that paper. So these concordance factors can kind of tell you for a particular gene or for a particular site, how many of your topologies agree with that. I can do this to everyone. Oops.
Don't worry, if you send it to me, I can repost to everyone.
Okay.
Oh, you did send it to everyone. So everyone,
okay.
Please, there is a link to what Dr. Dikow is saying on the chat right now.
So this implementation is actually, if you use the software IQ tree, which is a really a good maximum likelihood software, these gene concordance factors and site concordance factors by Min et al. is implemented there. So you can estimate for each gene or for sites to just better understand your data. And then you can kind of get a node, a branch support that has both the bootstrap, gene concordance factor, and a site concordance factor. And it's really interesting to see how they're just very, very, very different values. So, you can maybe just a single one of your gene trees contains a particular grouping, but maybe it's highly supported by a bootstrap. And so it's just kind of a different kind of information about your data that allow you to really think about which genes and which sites you feel are, driving the phylogenetic signal.
Thank you for that. Now, Manolo Perez has a question, and I had written the same question in my notepad, so thank you, Manolo, for that. He says, awesome talk, I agree. How can we try to be more inclusive with researchers from developing countries, thinking about the huge resources required for sequencing and analyzing huge data sets?
Yeah, I agree that this is a really important question. And, I have to admit, I'm not completely sure if the National Science Foundation funding clusters are available to outside of the United States. I think they are, but I'm not completely positive. But that's definitely something, it's a really big problem, especially with whole genome assembly and alignment, that these can take weeks. A genome alignment can take many weeks and hundreds of gigabytes of RAM. There's the potential for cloud resources to be part of this, but cloud can be really, really expensive. I know that Amazon Web Services does have a research grant program. So people can apply, I think, from anywhere in the world for a certain number of credits to analyze data in the cloud. That can be one option. Cyverse does have a cloud implementation, so that's an NSF-funded resource that researchers can apply for time on, and I think it's available to anyone. I'm not completely positive about that. So those are some opportunities. The Carpentries training program, which is not specific to genomics, but they do have resources in English and Spanish. So that's one step in the right direction. But I agree that this is not a solved problem. It's a huge problem and really important to put effort in.
Carolina Machado just said, Rebecca, it was a great presentation. Thank you. There is no question. I just wanted you to know. We have lots of those. Related to this question about making, let's say, the genomic revolution more accessible, more inclusive, to people in developing countries. I'm even imagining people in Brazil, where I'm from, in which I know it's not super accessible for people to be doing projects like this. Would you say that there is room for individually, if we're thinking about what individual researchers can do to help, are international collaborations a way towards maybe bridging this gap between people interested in maybe in poor research down there in Brazil and people interested in utilizing genomic resources and computer clusters in an efficient way in the United States? Is like thinking about creating those collaborations a way to go forward as well?
Yeah, I mean, I think absolutely, because yeah, I mean, I think collaboration is the only way that this work can really get done. I mean, it's just huge. I mean, there's not one kind of, one scientist that can kind of take on all these projects and have the expertise needed. So I think collaboration is really important. I think it's important, though, that it doesn't become like a helicopter science where,
Oh, sure.
people from the, US or other Western countries go to other places just to kind of get their samples and not give anything back. So I think that that's obviously a true collaboration should require that there be training and access to resources given to all parties. So I think that that's it's not solved by any means, but I think that's really important to consider.
Thank you so much for that comment. On a more technical thing, I'm seeing if there are more questions. I don't think I'm missing any. On a more technical question, you mentioned probes as one of the many ways that people are assessing a lot of data from different sources. It's very common to use probes for those that don't know what they are, I'm talking about something that you can enrich a certain different parts of the genome by looking to something that is very stable, but that's flanked by regions that change and that acquire information. And doing that with no model organisms is not uncommon. However, I wonder if what happens when we do not account for repetitive elements that may be flanking those ultra-conserved regions that are used for probes. Does your team has been doing any research on that? Do you have any opinions on that matter?
On how to capture those repetitive elements,
Or more precisely on how repetitive elements or repetition in the genome could hamper analyses that are based on UCEs, for example?
Yeah, I'm not sure if if there's been work done in that area. I think it's really interesting. But yeah, I'm not aware of it currently. I think it could have an impact. I mean, that's part of why there's always like this "it depends" answer, because you design your probes based on 2 or 3 maybe genomes, aligning them to one another, and then you try to amplify, enrich maybe hundreds of taxa that might be fairly distant. So it's always kind of probably a trial and error situation in terms of how your enrichments come, what loci end up being really easier to assemble and to align to one another. And so I think there's a lot of trial and error, and also specificity based on your taxa, with what works best. For the plant target enrichment project I'm involved in, the probes were designed from transcriptomes, but we're hoping to redesign them now that we have some more whole genomes, in order to really account for some of the more kind of genetic differentiation.
Yeah, that's a great answer. Thank you very much for that. Again, I reposted Dr. Rebecca Dikow's short bio and email in the chat so people can reach her easily. And I would like to remember you that at 2:50, we are starting Dr. Frederick Delsuc's question. If we don't have any other questions, I would just like to thank you once again, Dr. Dikow, for your time. I know you are a very busy person, and we couldn't be happier that you took your time to come here and honor us with your presence. Thank you so much.
Well, thank you so much for the invitation.
Thank you all to all the attendees, and thank you people at UNC Charlotte, Natasha and Marcos. You may close the room now. Goodbye to all.